Oh, hello. Carl Sagan once said that the secrets of evolution are time and death. Well, this statement is true. It leaves out the key element that evolution is built on, and that is heredity. The ability for organisms to pass on their traits from one generation to the next is a fundamental part of the evolutionary process. The true mechanism of evolution is the ability for those traits to change. In single-celled organisms, this change was mainly due to mutation, but the rise of sexual reproduction allowed for an explosion of diversity as the traits of the parents were no longer simply passed to the next generation, but they were rearranged, allowing for new combinations. As new genes and new gene combinations arose, natural selection was then able to select for the most adapted variations. This is the power of evolution, the creation and selection of new genes and novel gene combinations. At their most fundamental, genes are just long segments of DNA that can carry information. That information can be used to create other polynucleotides, such as RNA, or other polypeptides, like proteins. It was originally thought that each gene corresponded to an individual trait, such as eye color. While that is partly true, it is now known that genes can blend and interact in surprising ways. Genes are considered the most basic unit of heredity. DNA, in the form of chromosomes, is what is passed from parent to offspring, and it is the code that determines how that offspring will develop and function. We have previously looked at single-celled, non-sexual cell reproduction. Prokaryotes replicate by a process of division, and eukaryotes divide using mitosis. However, this process only creates a clone of the original. Sometimes mutations occur, but the divided cell is fundamentally the same as the original. This is called asexual reproduction. However, in sexual reproduction, genes are sorted into gametes, or reproductive cells, that can carry only half of the genetic information of the parent, which is then combined with the genetic information from the gamete of the other parent. As we saw last time, humans have 46 chromosomes in each of their somatic cells. Remember that somatic cells are non-reproductive cells and make up most of the cells of your body. Gametes in humans are the sperm and egg cells. They each contain only 23 pairs of chromosomes. This image is a karyotype, which is just an image of all the chromosomes from a single organism. Each somatic cell contains 2 sets of chromosomes. This means that each chromosome has a homologue. Homologues are chromosomes that contain the same genes, but the details of those genes are not identical. For example, you were born with 2 copies of chromosome 15, one from the egg and one from the sperm. Chromosome 15 is the location of a gene that helps determine your eye color. This is the HERC2 gene. However, the homologue of chromosome 15 from the sperm may carry a different variation of the eye color gene than the one from the egg. Yet each chromosome has those genes in the same place. This is called a locus. A locus is a place where a gene can be found on a particular chromosome and its homologue. So each copy of chromosome 15 has an eye color gene at the same locus, even if the details of each gene are slightly different. This is part of the variation from which evolution can select. You have a unique combination of chromosomes 15 that are distinct from your parents. So humans have 46 chromosomes, which are 2 sets of 23. Grizzly bears have 74 chromosomes, consisting of 2 sets of 37. Geneticists use N to represent the number of chromosomes in a haploid cell. So for humans, N equals 23, and for grizzly bears, N equals 37. A somatic cell contains 2N, or 2 copies of each chromosome. Last time we saw mitosis, which is the process by which cells duplicate their chromosomes and then divide, producing 2 daughter cells, each with 2 N chromosomes, the same as their somatic parent cell. However, gamete cells only have N chromosomes. These cells are created by a process called meiosis, which in many ways is just mitosis happening 2 times, but without a second DNA replication step. However, there are some important differences that we need to consider. Meiosis is broken up into 2 stages, meiosis I and meiosis II. In meiosis I, the chromosomes are replicated, and then condensed just as they did in mitosis. However, those condensed chromosomes do something different here. They participate in what is called crossing over, or homologous recombination. In homologous recombination, replicated chromosomes line up with their homologous pairs. proteins then facilitate the breaking and rearranging of those chromosomes. In simple terms, the chromosomes trade sections between each other. These are homologous pairs, so the sections that are traded contain the same genes. So if we look again at chromosome 15, we see that the copy of chromosome 15 that came from the sperm will swap sections with the chromosome 15 that came from the egg. This creates two new versions of chromosome 15 that are not exactly like what the cell started with. So let's take a look at meiosis step by step. Meiosis occurs in two stages, meiosis 1 and meiosis 2. Each of these steps contain phases that will be familiar from mitosis. However, they aren't exactly the same. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by cytokinesis. Mitosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by a second cytokinesis. Because the DNA only replicates one time, this results in the generation of haploid cells that contain only half the chromosomes as the original parent cell. In prophase 1, the nucleus breaks down and the chromosomes condense. This is a duplicated chromosome like we saw in mitosis. It consists of two sister chromatids bound at the centromere. Just like all chromosomes, this one is a homologous partner that is also replicated. During prophase 1 of meiosis, these chromosomes will cross over and recombine. They can do this because they carry essentially the same genes with closely related sequences. This allows for proteins to take genes from one of them and swap it with the same gene from another. Maybe brown eyes get swapped for blue eyes, or blonde hair gets swapped for black hair. Once they have condensed, a protein complex called the synaptoneal complex forms between the homologous chromosomes. Once the synaptoneal complex is fully formed, the homologous chromosomes are said to be in synapsis. This is where proteins can create breaks in DNA and attach those breaks to the matching site on the homologous partner. Once this process has occurred, and the synaptoneal complex has been disassembled, the chromosomes remain attached at what are called chiasmata. These are places where rearrangement has occurred. Metaphase 1 then occurs and the linked homologous chromosomes are lined up on the metaphase plate. However, instead of pulling sister chromatids apart, as was done in mitosis, the microtubules are pulling the homologs apart in anaphase 1. Then follows telophase and cytokinesis, much as in mitosis. However, we now have two daughter cells that are not identical copies. Instead, they have separated the homologous chromosomes and their sister chromatids into different cells. These two daughter cells can now divide again in meiosis 2. Chromosomes are still bound to their sister chromatids at the centromere in prophase 2. Again, the nucleus destabilizes and the microtubules grow from the centrosomes. In metaphase 2, we see the microtubules have bound to the centromeres of each pair of sister chromatids, and in anaphase 2, they are pulled apart, just like in mitosis. Telophase and cytokinesis occur, and now there are 4 daughter cells, each with only N chromosomes. It is important to notice that meiosis recombines and resorts chromosomes. Each one of the 4 daughter cells receives a random set of N chromosomes. This means that each sperm and egg don't contain an exact copy of the original organism's genome, but they contain a remix. This is called independent assortment, and depending on random arrangement, those daughter cells could contain many possible different combinations. This allows for an incredible amount of diversity from which evolution can select. The evolution of sexual reproduction has given organisms an increased ability to adapt to changing conditions through increased diversity. I feel like you can see the rest of the room in my glasses.